Well, good afternoon, everybody. Thank you for this opportunity to share my work. I'm really excited to be here. Before I get into my research, I want to just give a little bit about my background. I was born and raised in a small town called Yucaipa in Southern California. You probably never heard of it. It's at the foot of a beautiful San Gorgonio mountain range. I went to college at UC Riverside, which is not too far from Yucaipa. Got my bachelor's degree in biochem there. I then moved across the country to Baltimore, where I got my PhD in toxicology at Johns Hopkins University. At Hopkins is where I got my first experience studying the heart, where I had a project on autoimmune myocarditis. I liked studying the heart so much that I wanted to do a postdoc studying the heart. So I found a fellowship at Stanford where I was able to work on a research project on mitochondrial and cardiac physiology. I've been at Stanford for about 5 years now, 4 years as a postdoc. I was able to get a K-99, and I'm now appointed as an instructor there. I've been there now for about a year on my K award, and I'm actively looking for assistant professor positions. So I am now going to share some of my research. So the focus of my work is on mitochondrial dynamics, which includes several processes that manage the morphology and function of the mitochondria. So there are processes such as mitochondrial fission, fusion, biogenesis, and mitophagy. My specific work focuses on mitochondrial fission, which is a process of when the mitochondria can split into daughter mitochondria that are smaller and more spherical in nature. It's primarily going to be mediated by proteins such as DRP1 and its binding partners. Now, traditionally, mitochondrial fission has been thought to be pathological in the heart. There's been several studies done, one in particular here by Ong, that shows that if you induce ischemia in cardiac myocytes, that the cardiac myocyte mitochondria induce this mitochondrial fragmented state, which you can see right here. These mitochondria, if you study them, have decreased mitochondrial function, increased reactive oxygen species, and usually result in cell death. However, there's been some more recent studies coming out that show that mitochondrial fission may actually be beneficial. For example, one study done by Bernard has shown that if you silence DRP1, which mediates mitofission, you get a reduction in ATP production. This was confirmed in vivo. If you knock out DRP1 in mice, you get a reduction in the ATP levels. So what this suggests is that the processes of mitofission fragmentation are not always pathological. There might be some physiological role to this in the body. And this might be especially important to organs that are energetically demanding, like the heart. So one of the questions that I want to ask then is there a physiological role for mitochondrial fission and fragmentation in the heart? Now, to answer this overarching question, one of the first experiments that I did is, can fission and fragmentation exist in the heart independent of pathology? So to address this question here, I developed an exercise model to simulate energetic demands, physiological energetic demands in the heart. And what I found is that exercise does indeed induce this fragmented phenotype in the heart. If you put a mouse on a treadmill and have it run for 1 hour at 80% of its maximum running capacity, you'll start getting this fragmented phenotype shown here at the bottom. This is sedentary. These mitochondria are smaller, more circular. They also have there's more mitochondria and they're smaller area and perimeter. If you isolate the mitochondria and look at protein, what you'll find is that there is no change in the mitofusins, but there is an increase in DRP1, which indicates that this phenotype that we're getting is mediated by fission and not necessarily due to a lack of fusion. So since we were able to demonstrate that the existence of fragmentation in the heart independent of pathology, I hypothesized that these processes of fission and fragmentation may actually be necessary to meet the energetic demands, such as exercise. So what I wanted to ask is cardiac mitochondrial fragmentation required for maximal exercise capacity? So to test this hypothesis, I used a DRP1 knockout model that was specific to the heart. We treated mice with tamoxifen, and then we submitted these mice to a maximal exercise model, which is putting on a mouse on a treadmill and having it run at increasing gradation in speed. And what we found is that when you knock out DRP1 in the heart, you get a reduction in the maximal exercise capacity, shown right here. And if you actually look at the mitochondria, you get fragmentation with exercise, and this fragmentation is blocked when DRP1 is ablated. And not only is it blocked, the mitochondria are abnormally large. Some have these finger-like projections as if they're trying to fizz, but they just can't finish the job. So to finish, I just want to say that this is just a component of my work. I'm also trying to study the signaling pathways that actually direct this physiological fragmentation, and also what differentiates this physiological signaling versus the pathological signaling. I'm focusing primarily on the beta-adrenergic, AMP kinase, and calcium-dependent signaling, which are prevalent in disease models like ischemia, and I found are also being activated with exercise. And with that, I'd like to finish and thank my postdoc lab, also particularly Martin Kussman for all of his mentorship during the Keystone program, and particularly my funding, such as the Keystone Fellowship Program, for bringing me here to share my work. Thank you so much.
So I thought it was really interesting, and I'm a skeletal muscle physiologist, so I'm just curious, have you looked at DRP1 in skeletal muscle with exercise? Yes. It's not just the heart that exercises.
Absolutely. So skeletal muscle is also going to play an important role, and in fact, I've been doing parallel studies with one of my graduate students studying that, and it's actually going to be different based on the type of skeletal muscle that you're looking at. So for example, if you look at the difference between an oxidative and a non-oxidative tibialis versus the soleus, you may get some different responses. And actually what we see is that if you exercise in the tibialis, you actually start getting mitochondrial fusion, which is what a lot of the older publications looking at dynamics in skeletal muscle have been showing. However, if you look in a more oxidative muscle that's closer in morphology and function to the heart, as to the mitochondrial quantity, for example, like the soleus, you're not going to be getting as much fusion. And in fact, we didn't get as big of a change that we saw in the tibialis. The soleus, when you exercise, doesn't really have a change in mitochondrial morphology.
That was a nice talk. So as you know, DRP1 is phosphorylated before it translocates the mitochondria. So do you know what happens to DRP1 phosphorylation in your exercise model, and what might be the kinase that might be regulating that?
Yeah. So actually, there's several sites that the DRP1 can be phosphorylated, some that are activating, some that are inhibitory. I wasn't able to show this, but after 1 hour of exercise, you actually get a dephosphorylation of the 637 site, which is the inhibitory site. Normally, this DRP1 is phosphorylated, which keeps it out of the mitochondrial fraction. However, when you exercise, this phosphos site gets dephosphorylated. We think it's going to be through the calcineurin pathway, which the Lucas Serrano group has shown is very important. The calcineurin pathway is also activated with exercise through beta-adrenergic signaling.